Assalamu alaikum. In this video, we are going to cover two enzymes, SDH and GDH. Up till now, the ALT, ALP, GGT have been covered in the previous videos. So SDH stands for sorbitol dehydrogenase, and GDH, which is also written as GLDH, is glutamate dehydrogenase. Um, Before starting these enzymes, when we talked about ALT, we saw that in large animals, ALT is not a good indicator of hepatocellular damage. So, what we prefer in the large animals is um SDH, GDH, and GGT, in which SDH is the enzyme of choice, but it's unstable. So it's unstable and difficult to measure. So what we do is we also include GDH in the panel test, and also GGT, but their preference is like this to this, like most important one, second most, and the third most. So because of that, GGT has been discussed in the previous video. The other two are left. Now let's begin with GDH. Its levels increase in hepatocellular damage, particularly important in hepatic necrosis in horses and ruminants. The sample in which you can measure this is either serum or plasma. That makes a blood sample, and the anticoagulant you should be using is heparin, which means that it should be a green top tube. Why green top tube can be used in this case? Because these anticoagulants basically stop the process of clotting, and they have different mode of action. In heparin tube, the heparin or green top tube heparin does not interferes with zinc dependent enzymes like GLDH or SDH. You should remember, this GDH or SDH both are zinc dependent enzymes, and heparin does not interfere with these enzymes. But if we use purple top tube, which has EDTA, it will chelate the zinc, and so that the level of these enzymes are going to be falsely reduced in the sample, and that's why EDTA is not preferred for measuring these tests. uh, and this was all about GDH. The only thing you should remember is hepatocellular damage in large animals. Next we have is SDH. SDH is an important enzyme. Its levels are increased in acute hepatocellular damage in horses, but it's a very labile analyte because it is unstable. Its highest concentration are found in liver. So you can see it is present in hepatocyte, cytosol of the hepatocyte cells, and in that hepatocyte it causes the conversion of fructose to sorbitol, and it is a very specific indicator of liver diseases in all species. So you should know that not only in horses, it is also a good indicator in pets also. But if you talk about ART, it is good in pets only, but not in the horses. So um, it is a good indicator of liver diseases in all species. But you should remember one more thing, that not only because of liver diseases, but also because of other primary or secondary liver diseases, it can be increased. For example, many horses with inflammatory GIT disease will have high SDH, although it is not a liver disease. So you should know that. Also, it's important to know that SDH activity is going to be increased 24 hours or 1 day after liver injury. So if today liver injury has happened, after 24 hours you are going to see changes in the blood chemistry. SDH is enzyme of choice for detecting hepatocellular injury in horse and cattle, as it should be ALT, not AST, as ALT and LDH are not liver specific in large animals, but in pets they are. But SDH is not a stable enzyme due to its instability. GLDH or GDH, which we studied in previous slide, is more being more stable, is also included in our large animal chemistry panels. To measure SDH, you should have serum or plasma. Anticoagulant is again heparin, reason I have told you guys earlier. And interferences, like the things that will interfere with the sample, or sorry, SDH levels in the sample are sample being lipemic, hemolysis, or icteric samples. And those who want to see the detail on this thing, I have posted a lecture on like artifacts in the blood samples, and also on the vacutainers can watch those lectures for more details. So that was all about SDH and GDH. Those are liver specific enzymes detecting the or indicating the hepatocellular damage in the large ruminants. That was all about these enzymes. Thank you so much.